Okay, let's unpack this. We're delving into what is, really the grand mystery of how we or any primate get built.
Right.
We're talking about that very, very early embryonic development, specifically a critical stage called gastrulation. So if you've ever wondered how a tiny cluster of cells manages to lay out the entire blueprint for, your body, your spine, your brain, this is the deep dive for you.
It really is the critical window. If development goes wrong during gastrulation, which is when those 3 fundamental layers of the body get established, the consequences are severe.
I can imagine.
That's exactly why abnormalities at this stage lead to such a high incidence of early pregnancy loss or, serious developmental defects at birth. You have to understand the normal process before you can even begin to understand the pathology.
And for the longest time, that understanding has been, a black box.
Right.
Studying this stage in natural primate embryos has been almost impossible. Why is that? Why can't researchers just watch it happen?
There are just some major hurdles. For one, the technical difficulties in keeping an embryo alive and developing for that long in a lab dish are immense.
Okay.
Then there's the limited availability of embryos for research. And critically, there's the 14-day ethical restriction. That prevents us from culturing human embryos past the very beginning of gastrulation, around Carnegie Stage 7.
So that means that whole crucial period of late gastrulation, when the organ systems actually start to form, has been totally inaccessible.
Completely. The body plan is really being laid out between, say, day 14 and day 25, but we just couldn't look under the hood.
Until now. So enter our solution, the monkey embryoid.
Exactly. These are stem cell-derived embryo models, so they look and behave like a real embryo, but they come from pluripotent cells grown in a petri dish.
And our mission today is to analyze this huge breakthrough. Researchers have finally pushed these monkey embryoids past that day 17 barrier all the way to day 25.
Which is the equivalent of late gastrulation, yeah? Carnegie Stage 8 or 9. It gives us the first complete accessible platform to study this foundational moment of primate life.
And what's so fascinating is that this model could immediately help us answer a huge question in biology. Is building the body plan an intrinsic process? Do the cells have the pattern already programmed inside them?
Or does it rely heavily on external cues, like the support system from a placenta? Is it all about the pattern, or is it all about the fuel?
So to get to day 25, the team first had to master that initial phase, from day 7 to 17, where this simple clump of cells forms a foundational disc.
Right.
And that meant optimizing the starting material. They used something called low-passage naive monkey embryonic stem cells. Why is low passage such a crucial detail here?
Oh, it's a huge quality control issue. Think of it like this. Every time you culture cells, they divide. And over many generations, many passages, they can start to pick up genetic errors or, lose their potency.
I see. So they're not as robust.
Exactly. Using low passage cells means they were fresher, cleaner, and had their maximum developmental potential. And this one detail, which seems small, boosted the efficiency of forming the initial blastoids up to about 60%.
Wow.
They were just starting with the highest grade raw materials possible.
And the culture system itself needed a total redesign, right? They moved from a flat dish to an optimized 3D suspension culture. What does that actually mean for the embryoid?
It means it needed to grow in 3 dimensions without getting stuck flat on a dish. So after the initial aggregation, they moved the embryoids to a dish that was on a shaker.
A shaker.
Yeah. The key innovation here is that gentle, continuous movement. The agitation makes sure that developing cells get uniform access to nutrients, and crucially, it prevents them from sticking to the plastic, which would just stop their 3D development cold.
Okay, that makes sense.
And that system supported a much longer culture period. They got about a third of them successfully to day 17.
And by day 17, what was the structural evidence that they were on the right track? What does it look like?
It looked authentic. They formed that classic bilaminar disc structure. You can picture it as this tiny 2-layered pancake floating in the medium.
Okay.
And it had a very tight interface between the inner layer, the epiblast, which becomes the fetus, and the outer support structures, the amnion and yolk sac.
And the real signal that development was truly underway was the appearance of the primitive streak.
Absolutely. That groove is the first visible sign of gastrulation. Think of it as the construction crew's main entrance. It's the site where cells start migrating inward from the surface to create the middle layer, the mesoderm, and the inner layer, the endoderm.
And they saw that happening.
They did. They saw the streak start at the posterior end around day 13 and then move forward, which is exactly what happens in natural primate embryos at Carnegie Stage 7.
But the real ultimate confirmation came from the genetics, the single-cell RNA sequencing. What did that cellular census tell them at D17?
It confirmed they had a complete roster of all the required cells. So we didn't just see the structure. We knew exactly who was living in it. We saw the true stem cells of the embryo, the epiblast. We saw the cells actively diving inward at the primitive streak. All the necessary extra embryonic parts were there.
And they even found the precursors to our future sperm and eggs.
Yes, they even saw the successful induction of primordial germ cell-like cells, or PGCLCs, the absolute earliest precursors.
So we have the complete foundation. The disc is formed, the 3 germ layers are starting, and all the materials needed to build everything from muscle to blood to reproductive cells are all accounted for. That's a huge step.
It is, but day 17 is still just the foundation slab. Now they had to figure out the really monumental task. How do you get this foundation to start putting up walls and defining rooms?
This is where it gets really interesting, because that period from day 17 to day 25 is when the body plan is truly defined, and is where most of the previous models just failed.
Right.
They had to upgrade the lab environment again to support this huge growth spurt.
They did. The D17 embryoids, which were still these aggregates, were moved and cultured individually, and the medium they were in was significantly enriched. To use your analogy, they needed way more fuel.
So what did they add?
They shifted to an enhanced medium, and then critically, they later supplemented it with 50% fetal bovine serum, which is, just a rich, complex metabolic soup.
And that boost worked.
It worked phenomenally well. They got a really high success rate. 113 out of 220 of the D17 models made it all the way to D25. And almost 70% of those had the optimal shape.
So what did it look like? How did it change?
Visually, the structure went from being a simple, flat disc to an elongated, bent, and folding embryo. And that bending and elongation, that's the defining characteristic of Carnegie Stage 8 and 9.
So what were the key developmental milestones that started popping up between day 22 and 25?
Yeah.
This is the start of actual organ formation, right?
Yes, this is where the central nervous system begins. We saw the neural plate forming, which is that flat sheet of cells that will eventually fold up to become the brain and spinal cord. We saw the beginnings of neural folding, which created a neural groove-like structure. They also found the allantois, an extra embryonic structure, and really significantly, the formation of the primitive foregut and hindgut.
Meaning the embryo had successfully mapped out its digestive system from front to back.
Exactly. The anterior-posterior digestive axis was established.
That dynamic process sounds incredible. Did the 3D imaging give them any surprising insights into how that folding actually got started?
It did, yeah. The light sheet imaging clearly showed the elongation and the folding, and it visually matched the human CS8-9 stage. But they identified this key transitional phase around day 20 they called preneural folding.
Preneural folding.
Right. The disc flattened out and elongated, but it did so while maintaining its D17 curvature. It was almost like a pause, a structural preparation before the big architectural changes kicked in.
So development isn't just a sudden jump, it's a series of highly structured preparations.
Precisely.
Okay, so they've mastered the structure. Let's look at the cellular complexity, the assembly line that built it all. The analysis of over 100,000 cells gave them a complete map.
A stunningly complete map. They found 25 distinct cell clusters, representing nearly every single cell type you would expect at this advanced stage. We can look at a couple of key areas.
We'll start with the neuroectoderm, the future brain and face.
Okay, so the trajectory was perfect. General ectoderm cells transitioned into specialized neural plate-like cells, and then they branched into these highly complex border cells.
And border cells are crucial because
they differentiate even further into play-code-like cells, and more importantly, neural crest-like cells.
Ah, the neural crest cells. Those are the wandering stem cells of the early embryo, aren't they? They give rise to the peripheral nervous system, bone, cartilage in the head and face.
That's them. So they found the entire blueprint for these structures. But, and this is important, they hit a biological ceiling. The marker for a mature, fully formed neural tube, a gene called PX6, was absent.
And why is that important?
It actually confirms the model's accuracy. Natural embryos that are cultured in vitro also stall at this exact same point. It shows the limitation isn't in the cells themselves, but in the environment we're giving them.
I see. And what about the mesoderm, the layer that forms muscle, bone, circulation?
It showed spectacular diversification. It stemmed from the primitive streak and branched out into all the separate lineages: paraxial, intermediate, lateral plate. They even identified precursors to heart cells.
But there's a bud coming.
There is. The diversification was complete. It's like the cells successfully organized all the raw materials and specialized labor. But here's the critical roadblock. Despite all that specialization, the embryoids failed to engage in advanced organ morphogenesis.
What do you mean?
They made cardiac mesoderm-like cells, but no beating heart. They made precursor cells for the skeleton, but no somites. And again, this mirrors the exact failure point we see in cultured natural embryos. The blueprint is finished, but the large-scale construction just doesn't start.
But one system that did succeed spectacularly was the circulatory system, the formation of the first blood. Tell us about that, about the yolk sac hematopoiesis.
This was a complete success story. Traced from start to finish, they saw the full trajectory. Mesoderm progenitors became hemogenic angioblasts.
Which can make both blood and vessels.
Right. And those then matured into hematopoietic progenitor cells. From there, they observed the complete spectrum of mature blood cells. Monocytes, macrophages, red blood cells, even megakaryocytes.
So the model actually figured out how to build functional mature blood cells completely on its own in vitro.
Yes.
Yeah.
And the cell proportions shifted exactly as they do in nature. The earliest precursors were dominant early on, but by day 22-25, the mature blood cells were dominant. It's a fully functioning, self-organizing system inside the embryoid.
Incredible. And finally, the primordial germ cells, the PGCLCs. There's been this long-standing debate about where future egg and sperm cells actually come from in primates. What did this model show?
Well, this model offered some really compelling spatial evidence. They traced the PGCLCs starting from the nascent amnion cells. They then watched them migrate along a very distinct path from the amnion to the posterior epiblast, into the yolk sac, and finally to the hindgut.
So that supports the hypothesis that they have an amniotic origin.
It strongly supports it, yeah. And that's fundamentally different from the mouse model, where they originate directly within the epiblast.
Okay, so to recap this second phase, by day 25, the embryoids built a complex, elongated structure. They established the gut axis, created a mature blood, and confirmed the migration path for reproductive cells.
But they also hit these critical bottlenecks in neural and mesodermal organ formation.
Right, so the model is a magnificent success, but its limitations might be its greatest strength.
Exactly, because they illuminate the precise developmental requirements that are still missing from the lab dish.
So let's dissect those key differences and limitations, the missing parts. What failed, and what does that failure teach us?
Okay, so the first major failure was the trophoblast lineage. That's the lineage that normally forms the bulk of the placenta. Post D17, the trophoblast showed really poor development.
And why was that?
It's almost certainly due to the suspension culture. The cells just didn't get the necessary attachment cues they would normally get from a uterus wall.
But here's the interesting part. If the structure responsible for placental support and external communication failed, why did the internal embryonic development proceed so well?
And that takes us right back to our first question, pattern versus fuel. This success suggests that the primitive endoderm, which forms the yolk sac and was perfectly differentiated, appears to be sufficient to establish the entire body plan structure during gastrulation.
So the trophoblast's role then might shift in these later stages. It's not about establishing the pattern.
It's about providing the fuel and the stable environment needed for that pattern to grow. The blueprint seems to be intrinsic to the inner cells.
Fascinating. And what was the second key structure that failed to appear?
The node. The node is the embryo's organizer. You can think of it as the embryo's internal compass. It's vital for setting up left-right asymmetry, determining which side your heart forms on.
Right.
They found the precordial plate, which is the precursor structure, but the node itself, which you can identify by markers like nodo and nodo, was absent.
And why is that absence so informative? What does it tell us?
It highlights a very precise missing environmental requirement. In mice, the node and its precursors are very closely linked. But this monkey model supports the idea that in primates, these structures emerge separately.
I see.
The fact that the precordial plate formed but the node didn't tells us that the current culture system supports the initial cell migrations, but it lacks the specific mechanical or chemical cues needed for the node's complex morphogenesis, the physical folding and reshaping it needs to do.
Okay, so this platform is designed to be a powerful tool, allowing for manipulation you could never do in a natural embryo. And to prove that, the researchers knocked out some key genes using CRISPR.
Yeah, they focus on these master regulators of gastrulation, the T-box transcription factors. First, they looked at TBXT, also known as brachyury.
And knocking that out.
It severely impaired both mesoderm and endoderm differentiation, which confirms its essential conserved role across mammals.
And what about the impact on the body axis?
The embryoids had severely shortened discs and a scrambled front-to-back axis. The anterior HOX genes, the ones that build the upper body, were upregulated, while the posterior HOX genes for the lower body were downregulated.
So it validates that TBXT is crucial for defining that full antero-posterior axis in primates.
Without a doubt.
And the other knockout, EOMs, showed an even earlier role.
It did. The cells completely failed to even form a proper blastoid structure in the first place. They had a severe lack of cavity formation. This showed that eomas is required extremely early on, likely because of its role in early trophoblast differentiation.
So this monkey embryoid platform, it really is the first robust long-term model of primate-like gastrulation. It provides a map for understanding human body plan formation and, crucially, the origin of so many early developmental pathologies.
Its greatest utility is accelerating mechanistic discovery. We can now use tools like CRISPR barcoding to screen dozens of developmental regulators in a primate context. That was just pure theory a few years ago.
And because you can generate so many of these, it opens the door for screening drugs.
High-throughput screening for drugs targeting developmental disorders, exactly.
The success of this model is phenomenal, but as you said, its incompleteness is maybe the most instructive part. If the primitive endoderm is sufficient to lay down the entire body plan, as this model suggests by surviving without functional placental support, does the role of that external support system shift entirely from creating the pattern to simply providing the fuel?
And what is that minimum environmental cue? Is it mechanical tension? Is it a single missing signaling molecule that is still needed in the lab dish to unlock the formation of key structures like the node and full organ differentiation?
That's something profound for you to mull over as you contemplate the blueprints of life.